We often need a way to communicate evolutionary patterns from genomic data in the context of space and time. For example, we might want to understand how mutations in a specific protein like spike have emerged or transmitted through a specific country or region in Africa. Specifically, we could scroll down to our entropy panel and we could identify the site in the spike protein here that has the highest entropy, which is site 681. If we click on this to color by the genotypes at this site, we can get information about how these genotypes have emerged through time and also through space. However, each of the main Nextstrain panels usually shows information about time, space, and genotype, but not necessarily all of that information at the same time. For example, the tree shows genotype through phylogenetic structure, it can show time on the x-axis, and it can show genotype by color, but it can't show geographic information at the same time. Within the map view, we can see genotypes by color, and we can see geographic information by spatial position, but we can't see information about time. Similarly, for the frequencies panel, we can see information about genotype and time, but we can't see geographic information. So an important question here is, how can we effectively show the evolution at this site, 681, through space and time through different Nextstrain views? One way to look at this information altogether is to turn off the entropy panel and view the tree and map side by side in grid mode. When we do this and scroll up, we have a view that shows genotypes through time, space, and also through time here. We can additionally filter to the region that we're interested in, which is Africa, such that the map focuses on that region. This view gives us a high-level view of the patterns of these genotypes in space and time. But it's not easy to tell how these genotypes have played out in time at specific locations. To communicate more information about geography and the spread of these genotypes through time in a particular location, we can filter our data to a specific location. For example, if we're interested in seeing how these variants moved through Kenya, we can filter the data to just those from the country Kenya. When we do this, we get a map view that zooms in on Kenya. Our tree is filtered to only the tips that belong to Kenya, and our frequencies plot also changes to show only data from Kenya. Now we can see the frequencies of these different genotypes through time, through space, and in the phylogeny. This phylogenetic view is now somewhat redundant because much of the information that it communicates about time is present in the frequencies diagram. And the information that we need about genotypes is primarily also reflected in this frequencies diagram or in the map. We can turn off the tree in this case and keep just the map view and the frequencies panel. Since we're focusing on a specific country, we can also try to zoom in to a higher resolution for this map view. We can now reset the zoom, and this will take us to a Kenya-specific view showing division-level information where possible. We now have a country-level view of these genotypes at site 681. We also have a frequencies panel that's showing how these genotypes change through time. But in the map view, we're still looking at cumulative counts of genomes through time. This means this pattern reflects the final outcome at the current time point, but it doesn't show us how these genotypes have changed in these different locations at different times. One way to try to get at the information about what's happening at specific locations at specific times is to scroll up and use the date range slider to change the dates of the records that are being shown. We can click on this left handle of the date range slider and drag it all the way to the right to around June of 2021. As we do this, we can see that we're filtering records, and now we're only showing those data that correspond to the beginning of June. When we do this, we see that we only have a subset of records that have a genotype of H and R and none of the ancestral genotypes. If we scroll down to our frequencies panel, we can see that this has also been filtered to just 29 tips out of the 3,769 that were in the tree. From this view, we might get the sense that most of the data that were collected in the last couple of months were collected from divisions that were in the center of Kenya, somehow right in the middle of Kenya. This is a little suspicious because there doesn't appear to be a single major division here at this point, so we'd like to double check our data. We can do this by coloring the map by admin division information. When we do this, we see that all of the data points reflected in the map currently have an admin division of Kenya, which means there was no division level information in the tree or in the metadata, and so the map can only say that these samples were collected from Kenya. This is important because not knowing this information, we might have assumed that there was some specific outbreak happening in the middle of Kenya. Now that we can see that these strains don't have any division level information, we can reset our date range here and find that actually there are many different data points that have division level information. The color by here isn't very useful because almost every single division gets a color shade of orange. So even looking in the frequencies diagram, it's difficult to tell what's happening. But this does at least confirm to us that we have division level information that we can use to plot on this map. Knowing that we have division level information to plot on the map, we can go back to our original question of how are these genotypes changing through time at site 681 in the spike protein. So let's go back to coloring by that information. We can go to the color by menu. That's a different way to get to this information. We can select genotype. We can select S as the gene, and we can type in 681 as the site of interest. We're now coloring by genotypes at site 681 again. And now we can filter by date from a different perspective by dragging the right handle of the date range slider all the way back to the beginning of the time period. As we do this, we see that all the cases in the map disappear, and our frequencies panel becomes blank. For this example, the frequencies panel isn't helpful, so we're going to turn it off and focus on the map. We can now drag the left-hand handle of the date range slider to the right, and as we do so, we see that we're starting to filter in time windows to show data that corresponds to that window. As we do, the map will update its zoom points to show where different cases are appearing and different genomes are appearing through time. We can slide this manually to get a sense of what's happening in Kenya. And as we drag, we can see the occurrence of a new genotype popping up in the middle of Kenya, which we now know represents cases that don't have any division information. And as we drag, we can see these changes through time. We see new genotypes appearing, and then eventually these new genotypes fixing in most divisions. This can be a helpful way to explore your data, and it may be helpful in a narrative view to look at specific time windows, but this does require manual intervention on your part to set these different fixed time windows, and there's an alternate way to handle this in Nextstrain. To automate the filtering of this dataset in time windows using the date range slider, we can use the animation feature of Nextstrain. Click the reset button next to the date range slider to set the view back to all available time points, and then you can click the play button here. You'll notice that when we click the play button, this modifies the URL to include this animate information. This modification to the URL can be embedded in a narrative like any other URL. But you'll also notice that this date range slider is now moving at a fixed rate across time. And this allows you to animate the occurrence of these different genome appearances in different locations and see how different genotypes emerge through space and time. We can watch this play out, and this allows us to see how new genotypes, for example, appear on the eastern coast of Kenya and then completely replace the original ancestral genotypes. We can also change some of the parameters about this animation by setting the animation speed to slow or fast, but these controls don't modify the URL, so including them in your narrative won't have an effect on how it is displayed in the narrative. Now that we know how to animate the emergence of different genotypes through time and space, we may want to look at the spread of these genotypes in specific regions that have neighboring countries or neighboring divisions. For example, we may want to focus on the southeastern coast of Kenya. To do this, we can filter our data to inspect those regions, such as Kilifi, Mombasa, and Kwale. This now shows the cumulative genome counts for each of these regions across all time, and the URL has been modified to show those filters or reflect those filters. If we click play, we can now animate the occurrence of specific genotypes at these specific locations at different points in time. This view also modifies the URL to include this animate parameter that we can include in a narrative. By including this in a narrative, we can identify how ancestral genotypes are eventually replaced by these more derived genotypes that then appear to fix to the north, spread to Mombasa, and then eventually fix in these different locations. In addition to this map view, there are alternate views within Nextstrain that allow us to look at these data in a similar way, but from a different perspective. For example, we can reset this date filter now, and we can turn the tree back on, and we can also turn the map back off. And now we have only the tree view. From this tree view, we can select the scatter layout option. And when we do this with a color by for genotype, then we get this layout where the x-axis is showing sampling date, and the y-axis is breaking out the categorical variables of the different genotypes at site 681. By default, we're seeing branches here from the phylogenetic tree connecting the different points, which can be a little bit messy, so we can turn those off. And now we see just the data points for Kenya along that southeastern coast. And this view allows us to see that these ancestral genotypes, particularly ancestral genotype of P at site 681, was circulating from the beginning of April, that this H genotype first emerged around December of 2020, and then continued to circulate through June of 2021. We can also see that this R genotype has emerged more recently, but it hasn't circulated to quite the same degree in these locations as this H genotype. This is an alternate way of encoding information about time, space, and genotype by using spatial positions here in a graph to represent time and the genotype, and then using filters to focus on specific geographic locations. There are benefits and disadvantages to each of the views that we've looked at, and the view that you choose really depends on the question that you're trying to answer or the patterns that you want to communicate.